is also known as lactogenic hormone, lutetropic hormone, mammaliopin, and leuterotropis. Okay, it is produced by lactotropes, which are the nothing but eosinophilic cells of anterior pituitary. Okay, see, I will show here anterior pituitary. See here, eosinophilic cells of anterior. This is the anterior pituitary. Okay. Next, coming to the point. Okay. It is a protein with molecular weight of about 23,000, a peptide with 199 amino acids. It's with a peptide with 199 acids. Okay, amino acids. Okay. Let us know about prolactin regulation. Prolactin release inhibiting hormone of hypothalamus, which believed to be a dopamine, inhibits the secretion of prolactin. Okay, it inhibits the secretion of prolactin. This prolactin releasing hormone is increased due to during progeny lactation. Okay, this prolactin releasing hormone is increased during progeny lactation. Okay, the hormones estrogens and progesterone and oxytocin prolactin secretion. Okay, nipple stimulation, stress, sleep, and sexual intercourse promotes the prolactin release. Okay, the hormones, estrogens and progesterone, and oxytocin, prolactin secretion. Nipple stimulation, stress, sleep, and sexual intercourse promotes the prolactin release. Okay, did you got it? Next, coming to the point, biochemical functions. Prolactin is primarily concerned with the initiation and maintenance of location in mammals. Okay, prolactin is primarily concerned with the initiation and maintenance of location in mammals. Okay, this prolactin promotes transcription and translation in mammary glands. This prolactin promotes transcription and translation in mammary glands. It increases the level of several enzymes. This increases the level of several enzymes involved in the carbohydrate and lipid metabolism. Okay, next, prolactin promotes the HMP shunt pathway. HMP shunt pathway increases lipid biosynthesis and stimulates lactose production in mammary glands. It increases the lipid biosynthesis and stimulates lactose production in mammary glands. Okay, this prolactin promotes synthesis of milk proteins, casein, and lactalbumin. This prolactin promotes synthesis of milk proteins, casein, and lactalbumin. Okay, prolactin promotes the growth of corpus luteum and stimulates the production of progesterone. This prolactin promotes the growth of corpus luteum and stimulates the production of progesterone. Now let us discuss about glycoprotein hormone. These glycoprotein hormones possess certain structural similarities. Despite their functional diversity structurally, these hormones contain two subunits, alpha and beta. Alpha subunit is nearly identical, but biological specificity depends on the beta subunit. Okay. Mm, these glycoprotein hormones possess certain structural similarities. Okay, it possesses the certain structural similarities despite their functional diversity structurally. These hormones contain two subunits, okay, alpha and beta. Alpha subunit is nearly identical, but biological specificity depends on the biologically specificity depends on the beta subunit. Okay, the important glycoprotein hormones are thyroid stimulating hormone, follicle stimulating hormone, and luteinizing hormone. Okay, now let us discuss about thyroid stimulating hormone or thyrotropic hormone. This is produced by basophil cells of anterior pituitary and is glycoprotein in nature. These thyroid stimulating hormone are nothing but thyrotropic hormones produced by basophil cells of anterior pituitary and is glycoprotein in nature. This consists of alpha and beta subunits. It contains both alpha and beta subunits. The alpha subunit consists of 92 amino acids, while beta subunit have 112 amino acids. Okay, its carbohydrate content is both alpha and beta have several disulfide bridges. Its carbohydrate content is present in both alpha and beta, which have several disulfide bridges. Okay. Biochemical functions. Let us discuss about biochemical functions of thyroid stimulating hormone. This thyroid stimulating hormone binds to plasma membrane receptor and stimulates advantage cyclase with a consequent increase in cyclic AMP level. Okay, this cyclic adenosine monophosphate acts as a secondary messenger to mediate the functions. Thus, the thyroid stimulating hormone stimulates the synthesis of thyroid hormones at all stages. This thyroid stimulating hormone stimulates the synthesis of thyroid hormones at all stages such as iodine uptake, oxidation, organification, and coupling. Okay, it also enhances the release of stored thyroid hormone. Okay, it also enhances the release of stored thyroid hormone. Next, let us discuss about pituitary gonadotropins. The follicle stimulating hormone and luteinizing hormone are commonly known as pituitary gonadotropins. These tropic hormones influences the function and maturation of the testis and ovary. Okay, these tropic hormones influences the function and maturation of the testis and ovary. Next, discuss about these two hormones are glycoprotein with two polypeptide chains alpha and beta. The alpha chain is identical with 92 amino acids, whereas beta chain have functional specificity. Beta chain of follicle stimulating hormone contains 118 amino acids, whereas beta chain of luteinizing hormone contains 112 amino acids. Okay. Biochemical functions of follicle stimulating hormone. Let us discuss about the biochemical functions of follicle stimulating hormone. In females, follicle stimulating hormone stimulates follicular growth, increases the weight of the ovaries. It stimulates the follicular growth and increases the weight of the ovaries and enhances the production of estrogens. Okay, it enhances the production of estrogens. Whereas in males, follicle stimulating hormone, testosterone production required for spermatogenesis. Testosterone production required for spermatogenesis. Follicle stimulating hormone also promotes growth of seminiferous tubules. This follicle stimulating hormone also promotes the growth of seminiferous tubules. Okay. Next, biochemical functions of luteinizing hormone. Luteinizing hormone stimulates the production of progesterone from corpus luteum cells in females. Okay, this luteinizing hormone stimulates the production of progesterone from corpus luteum cells in females, whereas in males increases the testosterone production of um, Leydig cells. Okay, from Leydig cells. In males it increases the testosterone production from Leydig cells. Okay. Next, let us discuss about luteinizing hormone and follicle stimulating hormone are collectively responsible for the development and maintenance of secondary sexual characters in males. Okay, luteinizing hormone and follicle stimulating hormone are collectively responsible for the development and maintenance of secondary sexual characters in males. Okay, the next coming to the point, the pro-opiomelanocortin. Okay, pro-opiomelanocortin peptide family. Let us discuss about this. This family consists of the hormones adrenocorticotropic hormone, lipotropin. Okay, let's continue. This family, the pro-opiomelanocortin peptide family, as earlier I said, this family consists of the it consists of the hormones. um, uh, Adrenocorticotropic hormone, Lipoprotein hormone, and melanocyte stimulating hormone, and several neuromodulators such as endorphins and enkephalins. Okay, it contains about 24 amino acids, and it consists of several neuromodulators such as endorphins and enkephalins. Okay, this pro-opiomelanocortin peptide family contains the several neuromodulators as endorphins and endoenkephalins. All the members of mm, pro-opiomelanocortin, okay, are produced from a single gene into a single polypeptide. These all members of the pro-opiomelanocortin, and the members means they like this, okay? Adrenocorticotropin hormones, lipoproteins, melanocyte stimulating hormones, etc. Okay, are produced from a single gene. All these are produced from a single gene into a single polypeptide, approximately 28 amino acids. Okay, this pro-opiomelanocortin consists of three peptide groups. Okay, adrenocorticotropin hormone that can give rise to alpha melanocyte stimulating hormone, and corticotropin like intermediate low peptide. Okay, next beta lipoprotein hormone that can produce delta lipoprotein hormone, beta lipoprotein hormone, beta endorphin. The latter is endorphins and enkephalins. Okay, the latter is endorphins and enkephalins. Okay, this let's discuss about that family, which is for the classified. Okay, see here, pro opio melanocortin. Okay, is classified into adrenocorticotropic hormone and beta lipoprotein. This adrenocorticotropic hormone further classified into alpha melanocyte stimulating hormone and corticotropin like intermediate low peptide, nothing but CLIP. Okay, in shortcut we can say is CLIP. Okay, again I will say corticotropin like intermediate low peptide. Corticotropin like intermediate low peptide. Okay, next coming to the point, beta lipoprotein consists of delta lipoprotein hormones and beta endorphins. This delta lipoprotein hormone produces beta melanocyte stimulating hormone, and whereas beta endorphin contains with 31 amino acids produces gamma endorphins with 15 amino acids, alpha endorphin and enkephalin. Okay, endorphins and enkephalins are natural analysis that control pain and emotions. These endorphins and enkephalins are natural analysis that controls the pain and emotions. They bind to the same receptors as the morphine opiates and are believed to control the endogenous pain perception. These are believed to control the endogenous pain perception. Okay, beta lipoprotein derived from this beta lipoprotein derived from pro-opiomelanocortin, uh, which promotes by lipolysis and mobilization of fatty acids. Okay, the most important role for the formation of beta endorphin, it is used for the most important role for the formation of beta endorphins and enkephalins. Okay, next coming to the point, adrenocorticotropin hormone. This adrenocorticotropin hormone is a polypeptide with 39 amino acids. Okay, consists of 39 amino acids. The biological activity of adrenocorticotropin hormone residue in the first is 23 amino acids from internal end. It is with 23 amino acids from internal end. Biochemical functions of adrenocorticotropin hormone. Let us discuss about the biochemical functions of adrenocorticotropin hormone. This hormone is primarily concerned with the growth and functions of adrenal cortex. Okay, this hormone is primarily concerned with the growth and functions of adrenal cortex. Okay, this adrenocorticotropic hormone increases the synthesis of corticosteroids by the adrenal cortex and also stimulates their release from the gland. It also stimulates the release from the adrenal gland. Adrenocorticotropic hormone is found to increase the transfer of cholesterol from plasma lipoproteins. It is used to found. It is used to found for the increase or for for what purpose? Means to increase the transfer of cholesterol from plasma lipoproteins into the fascicular cells. Okay, fascicular cells. Okay, these adrenocorticotropic hormone activates mitochondrial enzyme cytochrome P450 side chain cleavage enzyme. This um this hormone activates the mitochondrial enzyme. This mitochondrial enzyme is nothing but cytochrome P450 side chain cleavage enzyme anamata. That converts cholesterol into um, um, pregnenolone. Okay, pregnenolone. Okay, this pregnenolone in turn is the immediate precursor of the synthesis of all steroid hormones. Pregnenolone in turn is the immediate precursor for the synthesis of all steroid hormones. Okay, it also activates dehydrogenases of HMP shunt pathway to increase the concentration of NADPH required for hydroxylation. It also activates dehydrogenases of HMP shunt pathway. For what purpose? Means to increase the concentration of NADPH required for hydroxylation. Okay. In adipose tissue, adrenocorticotropic hormone activates hormone sensitive lipase. Okay. In adipose tissue, this adrenocorticotropic hormone sensitive lipase, this enzyme is involved in the lipolysis. Okay, as earlier I said, in adipose tissue it involves the lipolysis, which increase the level of free fatty acids. Okay, next, adrenocorticotropic hormone has different effects on carbohydrate metabolism. It has different effects on carbohydrate metabolism. It lowers the blood glucose. It increases the glucose tolerance. Deposition of glucose in glycogen in adipose tissue is increased. The deposition of glycogen in adipose tissue is increased. Okay. Next, coming to the point, posterior pituitary. Okay, as earlier I said, um, posterior pituitary is formed from nervous tissue and consists of nerve cells surrounded by supporting cells called pituicytes. Okay, posterior pituitary is formed from nervous tissue and consists of nerve cells surrounded by supporting cells called pituicytes. Okay, these nerves, these nerves these nerves have their cell bodies in the these nerves have their cell bodies in the supraoptic and paraventricular nuclei of hypothalamus, and posterior pituitary secretes two hormones. Okay. Mm, as earlier I showed. Okay, I will show again. Wait. This is the posterior pituitary. Okay. Here you can see clearly. See here. This is the posterior pituitary gland. Okay. Mm, Posterior pituitary is formed from the nervous tissue. It is formed from the nervous tissue and consists of nerve cells. It consists of nerve cells. Um, Surrounded by supporting cells. Okay. These supporting cells called as pituicytes. Okay, these nerves have their cell bodies in the supraoptic, and paraventricular nucleus of hypothalamus. Okay, and posterior pituitary secretes two hormones, namely uh, as anterior pituitary. This posterior pituitary also secretes two hormones, antidiuretic hormone or vasopressin and oxytocin. Okay, oxytocin is synthesized from the paraventricular nucleus. Okay, this oxytocin is also synthesized from the paraventricular nucleus. See here, this is the oxytocin. It is synthesized from posterior pituitary. Okay. Next, coming to the point, an antidiuretic hormones is synthesized from supraoptic nucleus of hypothalamus. Antidiuretic hormones is synthesized from supraoptic nucleus of hypothalamus. Both of them are nanopeptides. ఇది రెండు కూడా nanopeptides with 9 amino acids. Okay. Next, coming to the point, okay, see here in chemistry, oxytocin structure. Okay, It contains nanopeptides. See here, 9 peptide bonds. Um, Here also, as earlier we discussed, here it contains two sulfide bonds between two cysteine bridges. Okay. Uh, See, in antidiuretic hormone also, it contains two sulfide bridges in between two cysteine bonds. Okay, next coming to the point, these two hormones are nanopeptides, contain 9 amino acids. Oxytocin differ from antidiuretic hormone with respect to third and eighth. See here, third and eighth. hmm, Third and eighth amino acid residues. Oxytocin contains isoleucine and third isoleucine, eighth leucine, and antidiuretic contains third phenylalanine and eighth arginine. The positions changes in both oxytocin and antidiuretic hormone. Here see in the third position it contains isoleucine and leucine. In eighth position it contains leucine in oxytocin. Whereas in the antidiuretic hormone, in third position we see phenylalanine, whereas in eighth position we see arginine. One intra-chain disulfide bonds is present between first and sixth. As earlier I said, two disulfide bonds are present. Intra-chain disulfide bonds are present in first and sixth. Okay, cysteine amino acids. The biological activities depends on C-terminal glycinamide. Okay, the biological activities depends on C-terminal glycinamide. Next, coming to the point, biological function of antidiuretic hormone. The main function of antidiuretic hormone is to conserve body water or decreases urine out. Okay, it is used to conserve body water or decreases the urine out. Okay, it has also same vasoconstrictor action. It has also same vasoconstrictor action. There are two types of receptors for antidiuretic hormone present in the body. V2 receptors at the collecting tubules of nephron, V1 receptors on the smooth muscle of anteriosus, V2 receptors at the collecting tubules of nephron. Okay. Okay, let's see. Very much. Okay. Okay, let's continue the class. V2 receptors at the collecting tubules of nephrons. Nephrons, okay. V1 receptors on the smooth muscles of anteriosus, okay. Combination of antidiuretic hormone with V2 receptors make the cell of the collecting tubule permeable to water. Whereas combination of antidiuretic hormone in high concentration with V1 receptor cause vasoconstriction. Okay, antidiuretic action. The main function of the hormone is to conserve body water. As earlier we discussed, the main function of the hormone is to conserve body water. Okay, the functions are mediated through cyclic AMP as secondary messengers. The functions are mediated through cyclic AMP as secondary messengers. Okay, the V2 receptors have strong affinity for antidiuretic hormone, whereas V2 receptors are present on the lining cells of the collecting tubular. Okay, these V2 receptors are present on the lining cells of the collecting tubular. When antidiuretic hormone combines with the V2 receptor, the adenylate cyclase is activated to form cyclic AMP. When antidiuretic hormone combines with the V2 receptor, the adenylate cyclase is activated to form cyclic AMP. Okay, the cyclic adenosine monophosphate acts on the cyst skeleton of these cells. Okay, the cyclic AMP acts on the cyst skeleton of these cells. Okay, ultimately this causes development of 5 intracellular channels within the cells communicating with the exterior, through which water passes from for reabsorption. Okay, this causes development of 5 intracellular channels within the cells. Okay, 5 intracellular channels within the cells communicating with the exterior through which water passes for reabsorption. Okay. Now let us discuss about vasopressin action. When the concentration of antidiuretic hormone is very high in the blood, the antidiuretic hormone causes arteriolar smooth muscles concentration. Okay. When the concentration of antidiuretic hormone is very high, యాంటీడయాటిక్ హార్మోన్ యొక్క కాన్సన్ట్రేషన్ ఎక్కువ ఉన్న బ్లడ్ లో ఉన్నప్పుడు యాంటీడయాటిక్ హార్మోన్ ఏం చేస్తుందంటే ఆర్టీరియల్ స్మూత్ మజిల్ కాన్సన్ట్రేషన్ కాజ్ చేస్తుంది అనమాట. దిస్ లీడింగ్ టు వాసో కన్స్ట్రిక్షన్ అండ్ ఎలివేషన్ ఆఫ్ ఆర్టీరియల్ బ్లడ్ ప్రెషర్. ఓకే, బ్లడ్ ప్రెషర్. Next, coming to the point, urea retention effect. Okay, permeability of medullary collecting ducts to urea is increased by vasopressin. Okay, the permeability of medullary collecting ducts to urea is increased by vasopressin. Okay, next coming to the point, and